Hi, I'm Gabby.
And I'm Kristen, and today we will be talking to you about gram-negative and gram-positive bacteria and different characteristics about each.
Okay, first we're going to get started on our model today. We would like to note that gram-positive and gram-negative bacteria have similar internal but very different external structures. The cytoplasm of the bacterial cell contains the DNA chromosome, RNA, the ribosomes, and metabolites. Unlike eukaryotes, the bacterial chromosome is a single-stranded circular DNA, and it's not contained in a nucleus, and it's free-floating in the cell, in the nucleoid. Histones are not required to maintain the conformation of DNA, and the DNA does not have nucleosomes.
Structure, components, and functions of the cell wall distinguish gram-positive from gram-negative bacteria. The cytoplasmic membrane of most prokaryotes are surrounded by rigid peptidoglycan, also called murin, layers. The exceptions are archaeobacteria organisms, which contain pseudoglycans or pseudomurins related to peptidoglycan, and mycoplasms, which have no cell walls at all. Because the peptidoglycan provides rigidity, it is also determining the shape of the particular bacterial cell. Gram-negative bacteria are also surrounded by an outer membrane.
Gram-negative cell walls are more complex than gram-positive cell walls, both structurally and chemically. Structurally, a gram-negative cell wall contains two layers external to the cytoplasmic membrane. Immediately external to the cytoplasmic membrane is a thin peptidoglycan layer, which accounts for only 5 or 10% of the gram-negative cell wall by weight. There is no teichoic acid or lipoteichoic acid in the gram-negative cell wall. External to the peptidoglycan layer is the outer membrane, which is unique to the gram-negative bacteria. The area between the external surface of the cytoplasmic membrane and the internal surface of the outer membrane is referred to as the periplasmic space. This space is actually a component containing a variety of hydrolytic enzymes which are important for the cell to break down large macromolecules for metabolism. These enzymes typically include proteases, phosphatases, lipases, nucleases, and carbohydrate-degrading enzymes.
In the case of pathogenic gram-negative species, many of the lytic virulence factors such as collagenases, hyaluronidases, proteases, and beta-lactamase are in the periplasmic space also. This space contains components of the sugar transport systems and other binding proteins to facilitate the uptake of different metabolites and other compounds. To differentiate, like we stated, this is our gram-negative bacteria model, but a gram-positive bacterium contains the same organelles, just not as many as this one.
Okay, and it's missing the outer membrane here, and just imagine having extra peptidoglycan layers up here.
So extra of the purple dots kind of stacked on top of each other and missing this part.
Okay. Positive bacteria has a thick multilayered cell wall containing many peptidoglycan layers surrounding the cytoplasmic membrane. The peptidoglycan of the cell wall is sufficiently porous to allow diffusion of metabolites into the plasma membrane. The peptidoglycan is essential for both the structure, replication, and survival of normally hostile conditions where the bacteria grow. During infection, the peptidoglycan can interfere with phagocytosis, is mitogenic, which means it stimulates mitosis of the lymphocytes, and pyrogenic activity, which induces fever.
The peptidoglycan layer, specifically in gram-positive bacteria, can be degraded by treatment with lysozyme. Lysozyme is an enzyme that is in human tears and mucus and is also produced by bacteria and other organisms. Lysozyme degrades the glycan backbone of the peptidoglycan. Without the peptidoglycan, the bacteria succumb to the large osmotic pressure differences across the cytoplasmic membrane, and they lyse, which means they destroy.
As mentioned previously, the outer membrane is unique to the gram-negative prokaryote. The outer membrane is a stiff, canvas-like sac around the bacteria. The outer membrane maintains the bacterial structure and permeability barrier of large molecules, such as proteins or lysozymes, and its hydrophobic molecules. It's Also provides protection from adverse environment conditions such as the digestive system of the host and heat.
Removal of the cell wall produces a protoplast that lyses unless it is osmotically stabilized.
The gram-positive cell wall may also include other components such as teichoic and lipoteichoic acids and complex polysaccharides. These molecules are common surface antigens that distinguish bacterial serotypes and promote attachment to other bacteria as virulence. Lipoteichoic acids are shed into the media and host, and although weaker, can initiate endotoxic-like activities. The outer leaflet of the membrane, which is the outside part, is composed primarily of amphipathic molecule, meaning that it is both hydrophobic and hydrophilic, called lipopolysaccharide, or LPS for short. Except for those LPS molecules in the process of synthesis, the outer leaflet of the outer membrane is the only location where the LPS molecule is found. LPS is also called endotoxin, a powerful stimulator of immune responses. LPS activates B cells and induces macrophage and other cells to release interleukin I and interleukin 6, tumor necrosis factor, and other factors. LPS can cause fever and shock.
The outer membrane of gram-negative bacteria also contains structural proteins and receptor molecules for bacteriophages and other ligands. The outer membrane is connected to the cytoplasmic membrane at adhesion sites and is tied to the peptidoglycan by lipoprotein. The lipoprotein is covalently attached to the peptidoglycan and is anchored in the outer membrane. The adhesion sites provide a membranous route for the delivery of newly synthesized outer membrane components to the outer membrane.
Now we're going to talk about another two structures that are similar to both gram-positive and gram-negative bacteria, the flagella and the pili. Flagella are rope-like propellers composed of helically coiled protein subunits, flagellin, that are anchored in the bacterial membranes through hook and basal body structures, and they're driven by membrane potential. Bacteria species may have one or several flagella on their surfaces and may be anchored at different parts of the cell. Flagella provide motility for the bacteria, allowing the cell to swim, chemotaxis, forward, or away from poisons. Bacteria approach food by swimming straight and then tumbling in a new direction.
So the fimbriae, which are also called pili, are these two structures right here. And they're hair-like structures on the outside of bacteria as shown. They're composed of protein subunits. The protein is called pilin. So these pili or fimbriae can be morphologically distinguished from the flagella, this longer one over here, because they are smaller in diameter and are not coiled in structure. So generally there's 100 fimbriae arranged uniformly over the entire bacterial cell, and they may be as long as 15 to 20 nanometers, or many times the length of the cell.
Fimbriae promote adherence to other bacteria or to the host. Alternative names are adhesions, lectins, evasions, or aggressins.
So you might be sitting there thinking, I don't know how to tell the difference between gram-negative and gram-positive bacteria. One important way that microbiologists are able to differentiate the two is through a technique called gram staining, which we do in lab. So as you can see from the diagram of the gram staining steps, the steps of gram staining in detail are...
Okay, so the first step is you're going to add the primary stain, which is also called crystal violet. The crystal violet will penetrate the peptidoglycan layer and stick to the cell wall. Now, the second step is to add your iodine, which is also called the mordant. This will help the primary stain to stick better to the bacterial cell. Then what you're going to do is carefully add a decolorizer. This is the differentiating part. Since the gram-positive bacteria has a thicker peptidoglycan layer, adding the decolorizer won't take all of the stain out of the cell, and the crystal violet will stick better. And since all of the stain is going to leave the gram-negative bacteria, since the peptidoglycan layer is so thin, you're going to add a counter stain, which is also called safranin. As you can see from the diagram, the gram-positive bacteria will stain purple. The purple is coming from the crystal violet, and since the peptidoglycan layer is so much thicker, the crystal violet can stain and keep the crystal violet after the counterstain and the decolorizer, which is ethanol, is introduced to the cell. Gram-negative bacteria, however, have a thinner peptidoglycan layer, so when you add the ethanol, which is the decolorizer, the crystal violet stain leaves the cell and adds the safranin better to it, which counterstains it pink.
Yeah.
And that's why gram-negative bacteria stain pink after the gram staining.
Yeah, so that's one important way microbiologists will, first of all, they'll look at their organism and whether or not, it's gram-negative or gram-positive, they'll know certain things about it, but the first step in discovering that is by this gram staining technique. And so the important thing to note is that a purplish or violet color is gram-positive and a pinkish color is gram-negative. I'm sure that most of you know that certain bacteria are harmful to our bodies. These bacteria are pathogenic, that's the term for it, and that can cause illness, diseases, anything minor to serious illnesses. Specifically for gram-negative bacteria, their pathogenic capacity is often associated with the lipopolysaccharide or LPS layer. So LPS acts as an endotoxin. And if an endotoxin enters the circulatory system, it can cause a toxic reaction. So symptoms of this include a high temperature, a high respiration rate, and low blood pressure. And in extreme cases, this could lead to endotoxic shock, which can be fatal.
Penicillin inhibits peptidoglycan synthesis. So therefore, the gram-positive bacteria that has a higher number of peptidoglycan layers is more susceptible to penicillin. And since the gram-negative bacteria, which is shown here, has a thinner layer of peptidoglycan, yet it has this protective outer membrane consisting of the lipoproteins, it is easier, or harder, I should say, for the penicillin to penetrate through and kill the bacteria.
So in general, it's more difficult to affect gram-negative bacteria with antibiotics, and that's why when you have antibiotics for a specific illness, whatever it is, the bacteria normally are gram-positive.
What we have here is a model of our gram-negative bacteria. The outer components of the cell differ, but the inner components are the same. Between both gram-negative and gram-positive. So they're both going to have the single-stranded circular DNA. They're going to have their extrachromosomal DNA in the plasmids. They're going to have their ribosomes. They're going to have what we have here in our yellow is our transport proteins. These are going to be the same.
And obviously the cytoplasm is this lighter green.
Yes. And the outer membrane is different. Both of them contain the purple, which is peptidoglycan. The thickness of these layers is different, but they're both going to have peptidoglycan. They're both going to have what we have here in blue is the flagella, and also back here what we have is the pili, which I don't know if you can see, but right here is the pili.
Also in blue.
Also in blue. The outer membrane is specific to gram-negative bacteria. The transport proteins in these are also yellow, but they're in the outer membrane, and they're called porin proteins, and they help pump molecules in and out of the periplasmic space, which is shown here in between the orange and the purple dots. And also the lipoprotein are shown in orange, and I showed the lipid bilayer here with the wires that are also shown in orange. So I hope our video has helped you differentiate between the differences of gram-positive and gram-negative bacteria. For instance, how gram-positive bacteria in the gram staining procedure stains purple, which means it retains a crystal violet.
And gram-negative stain pink because they accept the counter stain and don't retain the crystal violet since they have the thinner peptidoglycan layer.
While the gram-positive bacteria have the thicker peptidoglycan layer. In the gram-positive bacteria, we have teichoic acid,
which is absent in gram-negative.
And in the gram-positive bacteria, we do not have this periplasmic space.
Yeah, which is present in gram-negative only.
The lipopolysaccharides are absent in gram-positive bacteria
but present in gram-negative.
because they are present only in the endotoxins, while gram-negative bacteria have the exotoxin quality. Gram-positive bacteria are highly sensitive to lysozymes
And gram-negative have very little sensitivity.
because they can use the efflux pumps with these porin proteins and pump them out. Gram-positive bacteria are easily affected by penicillin, which we talked about before.
And gram-negative, as we discussed, is not very sensitive at all to penicillin, so that it's very difficult to affect a bacterial cell by taking antibiotics. So thank you for watching our little video, and we wish you luck in your future studies.
Go Gators!
Yeah!